Hello, everybody. Thank you for joining us again for the discussion on weed science and research. We're going to let the webinar room fill here for just a few seconds, and we'll introduce our speaker. Good afternoon, everyone. Thanks for joining us today for a discussion on weed science and research. I'd like to introduce Dr. Lee Van Wyken, WSSA's Executive Director of Science Policy, to start things off for us today. Go ahead, Lee.
Thank you, Eric, and welcome and greetings from Washington, D.C. As Eric said, my name is Lee Van Wyken. I'm the Executive Director of Science Policy for the Weed Science Society of America. As we say, weeds, we kill them, we don't smoke them. And my weed of the day behind me is Palmer amaranth, the biggest, baddest weed out there right now in agricultural settings. And fortunately, we're going to hear a little bit more about that later on. This is the 5th webinar in the WSSA USDA Agricultural Research Service web series, and we're very grateful for the leadership of Dr. Steve Young and Dr. Steve Duke for organizing this. Dr. Young has a monumental task ahead of him as the national program leader for invasive weeds or weeds and invasive pests, but he's doing a great job so far. He started just over a year ago and is providing the leadership that we've been missing for the last decade, I would say. So I can say that as a nonprofit scientific society, but we had a lot of issues out there for weed research. As we know, weeds left uncontrolled, just even in soybeans and corn, result in over 50% yield loss. That's scientific data over many years in many locations across the US and Canada. And that's $47 billion we'd have losses if we didn't control weeds. So there's a huge need for the weed research out there. And thankfully, Steve is doing that. So Steve is very qualified. He did his undergrad and master's in weed science and a postdoc out in California with Dr. Tomaso. He did stints in universities at Nebraska, at Cornell, and then at Utah State most recently until he came into this position. So it's an honor and privilege for me to introduce Dr. Steve Young. Thank you, Steve.
Thank you, Lee. I appreciate that introduction, and thank you for your support, WSSA as well. So again, really great to be here with everyone. Thank you for those who are joining us, and looking forward to hearing today. This is really exciting. We're like I think most of you have seen, we're kind of shifting gears. Last few weeks, we've been talking about the tactics, so tactics of integrated weed management. Now we're going to kind of go into the mechanisms, and today's going to be really exciting. We're going to hear about the role of plant physiology in weed crop competition from Dr. Dave Horvath. So Dave Horvath received his bachelor's from Purdue in 1984. Where he worked with Dr. Jeff Bentesen on identifying experimental conditions for various plant stresses as a base for finding mutations in corn that impacted the stress responses. Dave Horvath has received, Dr. Horvath received his PhD in 1993 from Michigan State University, working under Dr. Michael Tomashow, where he was part of the team that cloned the first cold-regulated genes from plants and correctly predicted the CBF. I'm not going to describe that. You can read his paper. I'm sure it's out there somewhere. CBF binding site in the promoters of cold-regulated genes using phylogenetic footprinting methods. So Dr. Horvath joined ARS in 1993 in Fargo, North Dakota, first as a postdoctoral research assistant to Dr. Lynn Dahlin, fine mapping the durable stem rust resistant gene RPG1 in barley before being hired permanently, thankfully, in 1995, where he was charged with identifying molecular targets that could be used to control the invasive perennial weedy leafy spurge. Dr. Horvath pioneered transcriptomics and genomics in weeds and helped identify dormancy-associated MADS, box. Again, you have to read the paper for that one. Genes that control endodormancy release in leafy spurge and other perennials. More recently, Dr. Horvath has begun studying the molecular mechanisms by which crops perceive and respond to weeds. And this is a really exciting topic. I think I've been watching it in the literature, and a lot of people are kind of pushing this along. So I'm excited to hear what Dave has to tell us today. So Dave, I'm going to turn it over to you. You can share your screen and tell us all about it.
Okay, let's see if I can get this right. All right, am I sharing my screen?
Yes.
All right. So I have to make a caveat before I start my talk. In case by some chance I zone out about halfway through or lose my voice about halfway through, it's because last night I was pretending I was 1/3 my age at the Who concert here in Fargo. So assuming that doesn't happen, let's see. Oops, and I've got to get back into this. There we go. Let's get to the slideshow. If I can actually do something with it. There we go. Assuming that I managed to go through with that, I really have to thank Steve and Steve Young and Steve Duke and the WSSA for giving me a chance to tell you about some of the work that we're doing to try to understand the physiology underlying why weeds reduce crop yield. Now, much of this work that I'm going to talk about today would be completely impossible if it were not for the work of a lot of ARS scientists, both past and present. So here's a group of the team that identified and isolated phytochrome in plants. And phytochrome is going to play a pretty significant role in some of the things that I'm going to talk about a little later in my talk. Additionally, I have to give shout outs to people at the Plant Gene Expression Center in Albany. These folks have helped develop many of the genomic tools that I was been able to leverage in my work in weed science, and the work, some of the developmental stuff that has been done by the likes of Sarah Hake on corn has really informed some of our results when we were trying to look at and understand how weeds reduce crop yields, particularly in corn. Likewise, there's quite a number of agronomists in ARS whose work I rely on. And most importantly, perhaps the group down in Morris, Minnesota, which Frank Frisella was in for quite a while. And if you remember Steve Duke's talk, he mentioned Frank. You know, without the agronomy stuff, informing and helping us choose the best model systems to work in, we would be lost. We really depend on the ARS agronomists to help guide us along our way. And of course, you know, there's tons of physiologists in ARS. The group at the Adaptive Crop Systems Laboratory are doing some really interesting stuff where they're looking at the physiology of weeds in response to climate change. And in part of that work is really developing some of the tools and databases that have helped me try to understand some of the data that we're seeing in our crop weed interaction studies. So getting into the meat of the talk here, we all know, so to speak, that weeds reduce crop yield. And it's generally assumed that the dominant paradigm is that they do so by competing with the crops for resources such as light, nutrients, and water, or perhaps producing phytotoxic chemicals or allelopathic chemicals that impact the crop growth, right? And that seems pretty self-evident, but I'm sort of curious as to how many people out here think that that's really the primary mechanism by which crops reduce, by which weeds reduce crop yield. So let's take a quick little poll here. And I'm just sort of curious how many people think that that's the primary mechanism, the competition for resources. Wow, this is awesome. Wow, quite a few people are voting too. This is great. starting to slow down a little bit. All right, we'll call that good. So right around 60, 66% or so of people think that this is a big, pretty much a true statement. Now, I'm going to spend the next 15, 20 minutes trying to convince you that there's a lot more to it. And in fact, that competition for resources, although it certainly occurs, is not the major reason why weeds reduce crop yield. Let's see if I can stop that. Okay, so now let's see if I can continue on. Get my mouse back over to my side of the computer. Uh-oh. Run into an issue. Let's see. Aha, there we go. There we go. Excellent. All right. So let's see if I can sort of convince you that there's a lot more to the mechanisms underlying how weeds reduce crop yields than just competition for resources. All right. So the first bit of evidence to suggest that there might be a lot more to it is a pretty well-known phenomenon amongst weed scientists called the critical period for weed control, or the critical, we'll go with that. And basically what that says is there is a specific time during the growing season where weeds have a really huge impact on yield. Now, if you get rid of the weeds before that critical weed-free period, you maintain pretty decent yield, 100%, close to 100% of your yield, right? And more interestingly, if weeds come in after that critical weed-free period, even if those weeds can get quite big and even sometimes even shading some of the smaller crops, there's very little impact of those weeds that come in later in the season, right? And the critical weed-free period happens really early in the growing season, usually from about, for corn, about the three-leaf stage to about the 8-leaf stage, or thinking about it from a developmental point of view or a developmental window point of view, really it starts when the crop plants switch from their juvenile phase to their adult phase. And it goes until that adult phase, until the meristems become floral competent at the end of the adult phase, and they start going into the reproductive phase. Now, for corn, they don't tassel until quite a bit out from this. But the meristems actually start producing tassel primordia right around the end of the critical weed-free period, about V8 in corn, actually about V7 in corn. So that's interesting that the critical weed-free period is so early in the growing season. And the reason that's interesting is because there's not a lot of competition for resources early in the growing season, right? So this is a graphic of nitrogen use in corn. And you can see that at the end of the critical weed-free period, right there between right about V8, that corn has only taken up about 20%, a little less than 20% of the nitrogen that it's going to take up to the rest of the growing season, right? So there's an awful lot of nitrogen left in the ground at the end of that critical weed-free period. And yet, that is the time point when weeds are having their greatest impact on crop yield. Now, you might think, all right, well, maybe what's happening there is when weeds are present, they're sucking up all that nitrogen really, really early and not leaving much left for the corn plant or the crop plant to deal with. But that also doesn't necessarily seem to be the case. This is a study, recent study that looked at the nitrogen accumulation in weeds fairly early in the growing season, when the corn was at V6. And I have to say that the weeds here in this particular study were having biomasses pretty close to the equivalent to the corn at the time. And yet, when you look at the nitrogen accumulation by the weeds, down there in the third column from the left, you can see that only about 3%, 3 to 4% of the nitrogen that was taken up out of the system was taken by the weeds and not by the corn. So it doesn't appear that the weeds are sucking up those nutrients very early and leaving none of it for the corn. In fact, most of the nitrogen that weeds take up is actually later in the season, well after the critical weed-free period. Now you might say, all right, so maybe if it's not nitrogen, maybe it's some of the other nutrients that are available or that the corn needs. And the picture that you get is fairly similar with other nutrients, with the exception perhaps of potassium, which seems to be taken up pretty quickly out of the system. But phosphorus, sulfur, again, nitrogen there showing that again. There's still generally quite a bit of it left when the corn is at the end of its critical weed-free period, and the weeds have already had all of their impact. So the weeds probably are not sucking up all of the nutrients and thus leaving very little there for the corn plants to use, and thus reducing their yield that way. The same is true for water, right? So water use really is the greatest towards the end of the season, in August or usually around in August, where you see a lot of water use. Early in the season, in that critical weed-free period, very little water is being used by the corn because it's not really all that big. And there's still generally plenty of water in the ground at that point in time. It's also probably not light. Here's a picture of an experiment we were running down in South Dakota, where we have corn growing with velvet leaf. And you can see that the velvet leaf there certainly isn't shading the corn very much. In fact, probably quite the opposite. Yet, shortly after this picture was taken, we removed those velvet leaf and looked at the yield of those corn at the end of the season. And those velvet leaf had already caused some sort of a change in that corn that reduced its yield significantly at the end of the season, right? And it almost certainly wasn't competition for light in that regards. Another hypothesis you might be able to come up with, if you were assuming that the primary mechanism for weed-induced yield loss is nutrient or resource competition, is that you should be able to replace that resource to the level such that it would negate the presence of the weeds, right? So if you have weeds there, they shouldn't be a problem with both the weeds and the crop have plenty of nutrients and water, right? But that doesn't seem to happen. Here you have a case here. This is a fairly old study, but it's very illuminating, where there's corn growing either with irrigation or without irrigation, and with or without quackgrass. And you can see that regardless of the irrigation levels, the water potential of those corn plants is not really affected by the weeds. Likewise, you can't just give them enough nutrients to compensate for the weed presence. Here's another, again, fairly old study back in 1967, but that's where all some of the really cool agronomic research was done fairly quite a while ago. And you can see that although weeds have a pretty big impact when they're present, and this is looking at corn height, and even though when you give them more nutrients, the plants will get bigger regardless of whether or not there are weeds present or not. The reduction in yield is not shifted by the addition of nutrients. So we had about a 40% yield reduction by the weeds when there was no nutrients added. And you have a 35% reduction when you've really given them all the stuff that they could possibly need, up to 200 kilograms per acre or pounds per acre of potassium and 200 pounds per acre of nitrogen, right? So even under luxurious conditions, yeah, you might move the needle a little bit, but you don't move it a lot. Here's another similar study a little more recent, at least a little bit more recent, corn growing with and without weeds, or I'm sorry, with or without, yeah, actually with or without weeds and with and without additional nitrogen. And this is the percent biomass lost to the weeds. And you can see that in almost none of the years or locations do you see a significant reduction in the yield loss when you add nitrogen. So you can't seem to compensate for weed presence just by adding more nutrients. Another prediction you might make, if you're assuming that weeds reduce yield primarily through resource competition, is that if you had twice the number of weeds or twice the amount of biomass of weeds, that you should have twice the loss of yield loss in your crop. And again, that doesn't seem to be the case. Generally, what you see is just a few weeds have a very significant impact on the yield of the crop. And as you add more and more weeds, it just sort of stays level or sort of just trails off at a stable state where the crop sort of maintains the yield regardless of the amount of weeds or biomass of weeds that are present. And that's something you wouldn't really expect if the weeds were primarily reducing the yield by competing for resources, right? You'd expect more of a linear response. If you doubled the amount of weeds, you'd reduce the yield of the crop by half. And it's not really anything to do with soil either, right? So you might argue, or one of the arguments might be that all right, so maybe it's not nutrients, maybe it's just the volume of soil that the crop plant has to access those nutrients, and the weeds might be crowding them out. But this has been studied quite a bit by ecologists, and they term this phrase the tragedy of the commons, because if you grow two plants in equal amounts of soil next to each other, you have no significant loss in yield. But if you put those plants in double the amount of soil in the same pot, all of a sudden you get a reduction in yield, right? So this is a study by O'Brien in 2005 where they did this, and you can see that the biomass during competition, even though the amount of soil was doubled, is still manifesting. And to be frank, corn plants don't need all that much soil to grow and live in. Here's a pretty substantial corn plant. This is one of our recent transgenic hybrids here that is growing in a 3-gallon pot. It's that's not a lot of soil. I can guarantee you those roots are taking up almost as much of the soil mass as I put in there originally, and it's doing fine. It's got three cobs on there that are full, and it seems to be doing pretty well in very small amount of soil. So it seems unlikely that it's competition for space as well, right? Now, sort of the nail in the coffin of this argument was done by a weed scientist, Clarence Swanton, who's up in Guelph, Canada, who many of you probably know. He knew about all these observations, and he wanted to test to see whether or not weeds could reduce a crop yield without any competition at all. So he grew a corn plant out in pots in the field and surrounded those corn plants by completely independent pots of a weed. I think in this case it looks like nightshade to me, but I'm not a weed expert. And as you can see, those pots of weeds are completely separate. So they've got their own separate drainage. There's no way that those weeds are going to be competing with that corn plant for nutrients. And that corn plant's going to rapidly, probably, although it's about the same size there as the weeds in this picture, you can bet in about a week that's not going to be the case, and that corn plant's going to be well overtopping those nightshade plants. And yet, even under those conditions, Dr. Swanton saw that those weeds had a negative impact on the yield of those corn plants, and in fact demonstrated that they had a critical weed-free period response as well. Right? So that all of this data to me says that it's probably not competition for resources. There's got to be something else that's happening here. So I'm kind of curious to see whether or not I've moved any of the 60% that thought that was the case. So go ahead and pop the poll back up again here. Let's see how many people I've managed to convince otherwise. Oh, look at that. I like the start. Oh, this is wondrous. This is better than I thought. Excellent, good. All right. Oh, we're pretty good there. All right. So look, I clearly moved the needle quite a bit. So I'll take that as a win. So if it's not competition for research, let's see. Let's see if I can get back. Let's see. I got to get back up here and I got to clear that. I've got to get into the other computer or the other screen. There we go. I've managed to do that. There we go. Now I got to get back into my computer with the screen. Nope. Come on, mouse. Go over to the other side here. Trixie. I can. Excellent. All right, I'm in. All right. So if it's not about competition, then there has to be something else there, right? So our current new paradigm that we're working under is that what happens is the crop plant perceives the weed, and once it perceives that weed, it starts some developmental changes that result in reduced growth, right? So why would a crop plant do this? Well, it doesn't make sense in a well-managed agroecosystem where there's probably plenty of nutrients around, but it does make sense way back in time in nature when plants might be growing in conditions that really were fairly nutrient poor, right? Or where they couldn't expect to see, a farmer go out there and throw nitrogen at them at prodigious amounts in the middle of the season. So plants have probably evolved to detect their neighbors, right? And once they detect their neighbors, they know that there's a possibility for competition. And therefore, they need to start conserving their energy, conserving their growth, and to make sure that they have sufficient nutrients available to them to get some seeds into the next generation and hope for better later on. Now, that's really kind of necessary for plants in the wild, but it's not really necessary for crops in a well-managed agroecosystem. And that opens up some really interesting possibilities, right? So if by chance we can understand what those signals are from that the crop is detecting from those weeds, it might be possible to block that response and then make the crop plant essentially ignore the weeds, because it really probably could afford to ignore those weeds in a well-managed agroecosystem and then just grow normal, right? So one of the major focuses of our research is trying to understand what those signals are and how they work in the crop plant, and then perhaps come up with a way to block that response and make crop plants more tolerant to weeds. So the first thing we did to try to get a handle on what was happening in the crop plant is we decided to take a transcriptomics approach to this problem. And basically the idea is that if you look at all the genes that are turned on and off in a plant when weeds are present or not, you should be able to get an idea of what is happening physiologically in that plant when weeds are present. And then from that information, maybe figure out what might be happening to cause those crops to make those developmental changes that reduce their yield. So we started doing this with microarrays and then switched over to RNA-seq studies because RNA-seq is much more accurate, I think, than microarrays in general. And And we were aided, fortunately, by Brian Scheffler's group in Stoneville, Mississippi, the Genomics and Bioinformatics group, because he was kind enough way back in the day to take the RNA that we obtained from an experiment here where we had weeds growing with or without soybeans over a 4-year period. And he kindly made the first RNA-seq libraries for us and sequenced them for us for free, and then helped guide me in some of the bioinformatics approaches to understand what that data meant. And we did that and looked to see which genes were differentially expressed when those plants were growing with or without weeds. And intriguingly, when weeds were present, we saw a downregulation, and I should say the orange colors, orange to black is downregulated, yellow to white is upregulated. We saw a downregulation of a large number of heat shock response genes. And that sort of confused us a little bit. We weren't really expecting that until we looked over on the right-hand side there and saw that PIF3 was upregulated. Now, PIF3 stands for phytochrome interacting factor 3, and it's involved in detecting changes of far red light, the ratio of far red light to red light. And PIF3 is known to both regulate heat shock responses, interestingly enough, and also regulate some of the shade avoidance responses that are important here, and I'll talk about them in a moment. Specifically, turning on some of the gibberellic acid responsive genes that are also in this upregulated gene set here. So, with PIF3 being upregulated, that gave us a pretty good clue of what might be happening, at least part of what might be happening, for understanding the mechanism by which those weeds were impacting the yield of those soybean plants. And what happens here is weeds or plants, chlorophyll, I should say, chlorophyll absorbs red light and reflects far red light. And plants, through the phytochrome and the phytochrome interacting factors, can detect that shift in the ratio of red light to far red light. And when plants do that, when they see an excess of far red light, it changes their developmental process, right? And it changes it in such a way that the plants tend to grow taller but more spindly. They put more energy into their shoot production than in their root production, and they flower earlier. And all of those things can have a pretty significant impact on crop yield, right? In addition, photosynthesis is impacted pretty significantly by excess far red light. It actually causes the photosynthesis systems to not work as well, and they start creating a lot of free radicals that result in oxidative stress that also can have a negative impact on plant growth and development. So one of our hypotheses, one of the signals that we think that the weeds might be giving to the crop plant is excess far red light, and that then changes their development. And I should say also that this is only really going to affect development if it happens at a time when the plants are still developing, right? So the plants have to perceive the signal prior to them flowering for there to be a major impact by this signal, much like what we saw with the critical weed-free period. Way back in 1993, another great weed scientist and former president of the WSSA that Steve Duke also mentioned, was studying how a cover crop impacted weeds, hairy vetch impacted weeds. And he noticed that weeds that were, the hairy vetch that was alive had a much bigger impact on the weed population, on the weed growth, than hairy vetch that was dead. And he hypothesized, although he didn't actually do the studies to prove it, that the reason that the live hairy vetch was impacting the weeds more was through the same sort of a system of shade avoidance, inducing a shade avoidance response in the weeds and thus reducing their growth and yield. So that's the story with soybeans. We were also interested in what happens in corn. So we did another study where we were looking at corn growing with or without weeds. And we did this in a 2-year study. 1 year we had them growing with velvet leaf, and the other year we had them growing with canola as a weed substitute. And the reason we chose 2 different weeds for different years was because we wanted to look at more of a general response. We know that, for instance, the canola is probably pushing out a pretty potent allele chemical, and we know that glucosinolates, the canola produces glucosinolates, which is a known phytoalexin. And we're pretty sure that the velvet leaf would not be producing that. So we didn't want to look for just something that was specifically responding to a specific allele chemical. We wanted to look at some more of a general weed response that would happen with any weed that was present. So that's why we chose 2 different weeds. And again, we did a transcriptomics study where we were looking at the response of those corn plants to weeds. And we got a little more sophisticated in our analysis here and did some gene set enrichment analysis on the genes that were being upregulated and downregulated in response to the weeds. And when we did that gene set enrichment analysis, we saw something that was pretty interesting. Now, the first thing that struck us was when we looked at the upregulated genes, they are highly overrepresented in functions involved in stress responses, specifically biotic stressors that are transduced through the stress hormones, salicylic acid, and jasmonic acid. We also saw that a number of other stress responsive hormones were upregulated. Citric acid and ethylene responses were upregulated. And in addition to that, we saw that phytochrome responses, just like in the soybean, were upregulated. And along with those phytochrome responses, we also saw indications of those sort of processes, the hormones involved in those sort of processes, such as gibberellic acid and auxin being upregulated. We also saw changes in osmotic stress responses being upregulated, which is kind of interesting. And intriguingly, we saw some indications of starvation responses, right? So that sort of went against our new paradigm hypothesis, as we saw some indications of nitrogen stress, nitrogen starvation, and phosphate starvation. Now, I have to point out that these plants were pretty well fertilized. So they should not have been short on these nutrients, but yet the plants were perceiving them as being perceived as being short on them. Likewise, when we looked at the downregulated genes, we see that the genes involved in photosynthesis are substantially downregulated when weeds are present. Likewise, genes that are involved in nitrogen transport, metabolism, and use are downregulated. And genes that are involved in growth and development are downregulated. And combined, when we see these stress responses being upregulated and these growth responses being downregulated, that sort of gave us another clue as to what might be happening in the crop plant when weeds are present. Whoops, I jumped ahead here in my talk. So in addition to the response of corn growing with weeds, and I should say that those plants were collected at V8 and the weeds were present all the way up through V8. We also took corn plants that we collected at V8 where we removed the weeds at V4. So sort of right in the middle of that critical weed-free period, to see what sort of processes might be changed sort of permanently by weed presence during that critical weed-free period. And I'll skip a little bit of this slide here and jump into the next one, just to focus in on the processes that were permanently impacted by weeds growing during that critical weed-free period. And what we saw was two things: the defense responses and the salicylic acid response was permanently induced when the weeds were present. The phytochrome response was permanently induced. Likewise, we still saw reduction in nitrogen transport and nitrogen use, and we still saw a reduction in genes involved in cell division and meristem growth being downregulated. And this is after the weeds were removed. So once those weeds are there, these responses seem to go on at least to V8, and we assume probably to the rest of the season. So what this did, so looking at this data, this is where we sort of thought that maybe this was giving us some sort of a clue as to how those weeds were impacting growth and yield. And that was because it looked to us like the plants were being induced to start a defense response and focus on that over a growth response. And this is something that's been studied in eukaryotic systems for quite a while. And we know that there's a key regulator of that decision that eukaryotic cells make to either focus on defense or growth. And that is the Tor Kinase Complex. And Tor Kinase Complex is a cluster of proteins that's really fairly ancient. It's present in every eukaryotic from yeast to humans. And what this complex does is it sort of is modified by a lot of different signals, nutrient status, sugar status, stress responses that might be happening, oxidative stress can downregulate the Tor Kinase Complex. Stress hormones such as salicylic acid are known to downregulate the Tor Kinase Complex. And perhaps maybe even some of those singlet oxygen process. Well, we actually know some of those singlet oxygen processes that are induced by the shade avoidance response can also downregulate this Tor Kinase Complex. And when the Tor Kinase Complex is downregulated, then it starts to shut down. It starts to phosphorylate or fail to phosphorylate key genes that regulate growth and development. Things like cell cycle regulation are downregulated directly by modifications, phosphorylation modifications by this Tor Kinase Complex. Processes in autophagy, where the plants are trying to conserve their resources, are downregulated. Meristem cell growth and development is downregulated. Nitrogen assimilation is downregulated when these things occur through actions of the Tor Kinase Complex. So this was looking at this. This is sort of another one of our hypotheses, is that the reason weeds reduce crop yield is because they impact this Tor Kinase Complex, and that reduces the growth and development of the crop plant. All right, so that was working under field conditions. We were also curious to see what would happen if we grew these plants in a greenhouse where we could completely control for the amount of water and amount of nutrients the plants got. So we did an experiment where we were growing corn and sunflower here, as you see, with or without weeds, and under ideal conditions. So these plants are watered daily. They're fertilized weekly. They're in a greenhouse with augmented direct overhead lighting. So there isn't much shading. The lighting from the sun is sort of diffuse in there, so there shouldn't be any real shading that's occurring, at least once the plants get well above the weeds, the height of the weeds. And yet, even under these conditions, those weeds knock the heck out of those crop plants, out of the corn and the sunflower. This is the data from the corn plants here. And you can see that even as few as 2 weeds in the pots has a pretty significant impact on the crop yield. And likewise, as you go 4 weeds or 6 weeds in a pot, you see a bit of a reduction, but it's more like that what we saw with the increasing the weed number and weed density. It sort of tails off, and you see less and less of a response by the crop plant to more and more weeds. But the real point here is that even as few as 2 weeds under ideal conditions still causes yield loss, right? Looking at the gene expression levels from those plants, we found kind of an interesting observation. Under greenhouse conditions, we did not see the same stress responses being upregulated, and we didn't see the same phytochrome responses being upregulated. We did see a fair number of transporter genes being upregulated, which we're not really sure what that means, but pretty consistently, those experiments that I was just talking about were repeated. So a lot of RNA-seq libraries were run for this study. So we're pretty confident in this data. But when we look at the downregulated genes, there we start to see the same sort of output that we would expect from that Tor Kinase complex still being manifested. So we see downregulation of nitrogen use and transport, and we see pretty strong downregulation of genes involved in plant growth and development under these greenhouse conditions. So that sort of leaves us with a little bit of a question, because we were pretty convinced from the field data that we had a good handle on what was happening. We thought that, it was stress that was signaling the downregulation of the Tor Kinase complex. But now from this greenhouse study, there's clearly some signals that are getting to those crop plants that seems to be impacting the same output that we would expect from the Tor Kinase, but it doesn't seem to be the same impact. So now we're thinking that perhaps maybe there are other signals, at least under ideal greenhouse conditions, that might still be impacting the Tor Kinase complex, but we don't really know how. So to sort of get an idea of what that might be, we decided to take a slightly different approach. And that is, we wanted to identify some weed-inducible genes, genes that were always turned on when weeds were present in those corn plants. So what we did is we took a look at our field studies. And again, remember, this is 2 years of studies under field conditions. And we saw a fairly small number of genes that were consistently differentially regulated in both years of the field study. 25 to be exact, 6 that were upregulated and 19 that were downregulated. And that was interesting. So then we wanted to look at how many genes or look at the genes that were up and downregulated under our control conditions in the greenhouse. And we saw 170 genes that were upregulated by weeds, regardless of the number of weeds that were present, from 2 to 6, and in both runs of the experiments. We had 170 genes there. And then we looked to see what sort of overlap there was between the upregulated genes in this study and the upregulated genes in the field study. And we found 2 of them that were upregulated. The one that we're going to focus on is this DC1 nucleoredoxin gene. And the reason we're interested in that is because in a number of other studies, we saw that this gene happens to also be upregulated in both the leaves and roots in response to weeds. And it also happens to be upregulated fairly quickly within 7 days of the corn plant being exposed to weeds. So we're focusing on this gene. And we alluded to some phylogenetic footprinting in my little profile at the beginning there. So the idea here is that genes that are related and are regulated the same way should have sequences that control that regulation that will be the same, that will be conserved through evolution. So taking a look at the promoter of this nucleoredoxin gene from corn, we were then able to get the nucleoredoxin, the same gene, from sorghum and look to see what sequences were common between the the promoter of this gene and the promoter of the sorghum gene. And those regions there that are highlighted in yellow are the sequences that are the same. And then we also looked at another species, Saccharum, and looked to see what overlap there was between Saccharum and corn and sorghum, and those are the green highlighted regions. So those are the regions that we think might be the most interesting and which are most likely to play a role in the regulation of this gene. Now, to be fair, I didn't actually at this point know that this gene or this promoter sequence was going to be capable of turning a gene on in response to wheat presence. So we made some constructs. We took that sequence, that 3,000 base pair sequence, and we put it on two reporter genes, RED3 and NOD-G. RED3 is just a gene that will turn the plant pink under a particular wavelength of light. And NOD-G is a gene from Pseudomonas, which reduces or which degrades salicylic acid. So we had some hopes that that might actually expression, that gene under the control of the nuclear rhodopsin promoter might actually make the corn plants more resistant to weeds. And then we looked to see, we made the transgenic plants and looked to see whether or not those reporter genes were weed inducible. And fortunately, yes, they are. So I'll just say that it's about a twofold induction with weeds responsive in both of those reporter genes. And that's important because that means that we've got a sequence from a promoter that is truly weed inducible. So now we can go back in and start picking that promoter apart and trying to identify the sequences within that promoter that are responsible for this weed inducible gene expression. On the sad side, at least under greenhouse conditions, that NOD-G gene does not really have much of an impact on the ability of that corn plant to respond to weeds. So, or doesn't reduce the yield loss caused by the weeds. But we haven't looked at that under field conditions, and we really don't know for sure, even if that NOD-G gene is functional in these corn plants and is actually reducing the amount of salicylic acid. So we've got quite a bit of work to do there. In the minute here I've got left, conclusions. I think we still have a reasonable new paradigm for how weeds reduce crop yield, and that is by them generating a signal that is perceived by the crop plant, and that the signal then changes the development of that crop plant through changes in gene expression that we can then map back and try to figure out how they're doing that so we can block that response and make those plants more tolerant to weeds. And we think that's doing that through the Tor Kinase signaling and also through phytochrome signaling. And of course, we have a weed inducible promoter now that we can start to pick apart some of the specifics and identify some of the key transcription factors that might be involved in this signaling process. So where are we in the future? Of course, we want to test some of these NOD-G expressing plants in the field. That's going to be a bit of an issue, but we're going to see what we can go after with that. We want to alter that Tor kinase signaling because that's really key to our paradigm. And I think it's a testable hypothesis that we can make by knocking out or overexpressing some of the Tor kinase complexes, or looking at some of the changes in phosphorylation that are known to be impacted by Tor to see whether or not weeds have a direct response on the activity of that complex, and maybe use that to help us create weed tolerant crops. In addition, using that weed inducible promoter, we want to refine that a little bit because right now it's more of an more on situation as opposed to an on off situation. And we'd like to be able to identify those transcription factors that bind to that promoter and maybe design a much more powerful weed inducible promoter that we can then use to perhaps turn on genes that might make the corn plant more resistant to weeds, maybe turn on some allelopathic genes that really turn them on strong only when weeds are present, or maybe even put them into some sort of a cool reporter system so that growers can fly their UAVs over their field and look for plants that glow, perhaps in the middle of the night, and see, where the weed problems are likely to be starting in their field nice and early, so they can go out and nail the weeds and reduce that problem. So, with that, we'll take questions. It looks like I've got at least 5 minutes here to do that. So
yeah, thanks, Dave. That kind of took me back to my plant phys class in college. So thanks for the refresher there. It's a lot to swallow. We do have several questions in the Q&A. So if you stop sharing,
let's see if I can do that.
Yeah, it might be easier for you to.
Oh, I did. All right, there we go. Aha,
Yep.
Look at that.
Yep.
All right.
So a lot of
go to the chat.
Yeah, a lot of good questions. So
let's see. What do we got here? Let's see. All right. So we'll go with this first one here. Thanks for the informative presentation. I guess the common question around light quality induced crop weed competition is why this does not happen between the crop plants itself. I mean, the interspecific light competition, light quality induced competition. Actually, it probably does. But there are probably some other, it's a complex system. And clearly, crops, like for instance, corn growing at double density. We know that corn, when it grows at double density, and Marty Williams referred to some of this in his talk on the second session. When they do that, I think there's some other systems that sort of compensate. They sort of recognize their neighbors, right? And they can tell self from non-self. In fact, we know that plants can not only tell whether or not it's the same species growing next to them, but they can actually tell whether or not they're related individuals. There's some studies to show that they can, the ability of crops to recognize their neighbors is really acute. And so chances are what happens, and I'm just guessing here, but I think that it would probably check out, is yes, they see that there's another plant nearby and they go to respond, but then there's other signals that go, wait. This plant's an okay one. Sort of tamp down that response a little bit, you're all right. And they change the way the plants respond. And in fact, we did some of these studies as well as in similar ways that Marty did, looking at the response of corn, the velvet leaf versus corn to corn, way back in the days when we were still doing the microarray work. And what we found was kind of interesting. Corn plants growing with corn at double density altered their the photosynthetic response. I'm blinking on the name of the enzyme that was the key to that. But when corn was growing with corn, it was upregulated, and when corn was growing with velvet leaf, it was downregulated. So they can actually tell which neighbors they have and respond appropriately to those neighbors.
Yeah, so that sounds, and there's several questions in the Q&A about if the crop can see the weeds, see the weeds, right, and sense them. And so it sounds like from your work and others that they're getting these cues that maybe they don't know exactly, like should we test the corn? Can it identify, what species of weed that it's interacting with? But I mean, there's different. Yeah, right. There's differences in those cues. So that's really, it's kind of some really interesting work. So Dave, if you want to scroll up, we had a couple questions from Claire.
And I've lost my, oh, there it is again. All right. Good. Okay.
She's talking about,
I lost it again.
Yeah, she's talking about intercropping.
Can you read the question? Because I'm still having trouble with my mouse here, getting it over into the other screen.
Yeah. She's talking intercropping. She says, are there mixed results? It says there are mixed results from different papers with the same two plants, and I'm wondering what you think would be the main culprits for different results. And that's the results she's talking about: yield hit versus yield increase. So
So yeah, in some cases, and I'm going to guess at the exact point of the question here. There are some cases where there's mutualism that occurs, where the weeds might actually have some protective impacts on the crop. I know we did some studies with the Arabidopsis in the growth chamber one not too long ago, and we had a heat event where the temperature in the greenhouse went sky high. And it turns out that the Arabidopsis growing with the weeds in that case were actually a little bit protected. I'm going to guess that it kept them from drying out, sort of kept the moisture right around the base of the soil a little bit better. So yeah, there are certainly environmental factors that can impact the response of the crop to the weeds. And genetic factor. I mean, it is going to be a complex system. It's not going to be a simple answer.
Yeah. There's another question about the John's asking, so why do weed impacts vary so much from one year to the next, even if the timing, types, and density of weeds are similar?
Again, there's a lot going on, particularly in the field conditions, that can impact the total response. That Tor Kinase Complex, right? It really senses a lot of different inputs and relates those inputs into an output. So anything that tweaks it one way, there might be other signals coming in and tweaking it the opposite, right? So in one year that might be, such that it causes a major response, and other years it might, there might be other factors out there, humidity, the species of weeds, who knows, right? That can be impacting it.
Yeah. So we've got a couple minutes here left, and so Greg's asking, so what crops and weeds will you expand this testing to? Right now we're looking at expanding it to Arabidopsis, to be honest, because Arabidopsis is a great model system. And we've done a good bit of work to show that Arabidopsis responds just like a crop. So it shows a critical weed-free period, just like crops do. And it's a nice manipulatable system. And there are lots of mutants in Arabidopsis that we can use. Mutations in Tor Kinase, for instance, already exist. I don't have to make them. I can go out and look at them, just order them on the internet. So that's one of the things that we're starting to focus on a little bit. To try to get a better handle on this. And we're still quite interested in corn. Corn is probably one of still one of our major crops that we want to look at. And we're expanding into sunflower as well right now. So those are the three big ones that we're really focusing on: Arabidopsis, corn, and sunflower. I mean, it still seems like it's kind of early in the research, but yes. I'll just kind of maybe end on this question is, do we think that the sensing is potentially mediated by soil microbes? So yes. I didn't hear you touch on that, but I didn't touch on that. Thank you for that question. That's a great question. And yeah, I would be shocked. I would be totally shocked if we did not see soil microbes playing a role in some of the signaling. Microbes both, they're going to absorb some of the chemicals that plants put out, and then they're going to change them, and then they're going to spit them back out into the environment too, right? So, and we know that, just having different species there changes the species of microbes that are present. So it would be almost inconceivable to me to find out that microbes did not have some role in some of the signaling. We know that they play a pretty significant role in long distance signaling between trees, for instance, right? So we know that trees are actually detecting what's going on in their neighbor trees through mycelium and fungi that are touching both trees. So yeah, it would be highly unlikely that they would not be playing a role.
But
Wow.
we haven't tested that yet. We are working on that,
Yep.
as are
Great.
others.
Great. So I really appreciate this, Dave. And I know there's many other questions here. So as always, you're welcome to follow up with Dr. Horvath or
Please.
myself. Contact me, and I'll get you in touch with David. And yeah, I'd love to get your questions answered, but just not enough time at this webinar. But again, thank you, David, for your great presentation this afternoon. Really great to hear. And I want to just tell everyone, as I've done the last few weeks, we're going to shift gears a little bit. We're going to move out of cropping systems in a way and go to invasive plants. Next week, we're going to hear about the molecular basis for controlling invasive plants by Dr. Matt Tanko. So I'd invite you all to come back. Same time, same place. We'll talk some more about weeds and really get into this topic. So again, thanks everyone for joining us today.
Thanks.
And we'll see you next week.
All right.
Thank you, Dr. Horvath.
Thanks.
Need to get
Thank
you
you.
some more money.
Indeed.